Hello everyone, thanks for being here. I am really grateful for this opportunity. In this talk, I will share with you work I've done in the context of my PhD project, focusing on the question of this symposium, which is PET imaging of brain connectivity, hype or future. As you surely know, to study in vivo human-brain connectivity through neuroimaging techniques, we typically inspect a simplified version of the brain, modeling it as a network, where macroscopic regions are related to each other by links or connections of certain weight. The nature of those links depends on the aspect of interest as well as the imaging technique. This way, whole brain function and structure can be represented by a network graph or by its adjacency matrix and can be characterized on different brain conditions. In the last decades, most brain connectivity studies were based on functional MRI, as you can see in this plot based on a PubMed search done in March 2021. It indicates that our current perspective of brain function disproportionately relies on a single point of view or parameter, which is the Bolt signal, what might restrain our understanding of brain function, and with that, our overall perspective of the brain connectome. On the other hand, positron emission tomography, as you can see, is one of the least exploited techniques in the context of brain connectivity. Even though PET was the first technique proposed to study interregional relationships in the human brain and provides a more direct measure of neural activity than Bolt signal. This is partially because it involves the use of ionizing radiation and also because of the stationary nature and the poor temporal resolution of FDG PET measurements that hinder the estimation of interregion correlations at individual level. In most cases, metabolic connectivity is assessed through a correlational analysis carried out on a subject series. This approach relies to some extent on the assumption that the process being studied is coherent across different individuals. Besides these limitations, metabolic connectivity has progressively shown potential for clinical applications, and alterations in metabolic connectivity have been reported in several pathologies such as schizophrenia, epilepsy, Parkinson's, and Alzheimer's disease. However, little is known about the mechanisms underlying metabolic connectivity, and important aspects remain unclear. For example, to what extent does structural connectivity underlie metabolic connectivity? In the present work, we ask to what degree structural connectivity derived from diffusion-weighted imaging overlap with fMRI functional connectivity, metabolic connectivity or FDG covariance, and structural covariance in the healthy human brain. We treated the structural connectivity as a reference because it is indicative of actual anatomical connections, it has a good reproducibility, and it can be obtained at a single subject level. To address this question, we analyzed data from simultaneous PET-MR acquisition in a cohort of 56 healthy subjects. The acquisition started 30 minutes after injection of about 100 megabecquerels of FDG. The MR protocol lasted about 30 minutes and included T1, diffusion, and resting state fMRI sequences. And we used the first 30 minutes of least mode PET acquisition to reconstruct one PET image per subject. For all modalities, we used the same gray matter parcellation that was derived from a AL2 atlas, combining and removing some regions conveniently in order to reduce the number of small regions to minimize the partial volume effect in PET images. So we finally worked with 106 regions, including cortex, subcortical structures, and cerebellum. Regarding the image registration, instead of normalizing all images to a common space, we work in the native space of each subject to avoid the image manipulation and to better control the performance of nonlinear registrations. We constructed structural connectivity networks from diffusion-weighted imaging, and for that, after visual quality control of the images, we corrected them for intervolume movement, eddy currents, susceptibility-induced distortions, and signal dropout. Then, we fit in each voxel a multi-compartmental diffusion model called ball and sticks, and after that we perform a probabilistic tractography using a white matter seeding approach. As connectivity weight between two regions, we use the number of streamlines normalized by the surface of the region to take into account that the larger the region, the more likely it is to be reached by a streamline. And as a result, we obtain a symmetric connectivity matrix for each subject. So to construct a group structural network, we resampled the connection weights of each subject to a Gaussian distribution. Then we calculated the mean structural connectivity weight of each connection across subjects. And finally, to minimize the number of false positives, we excluded those connections that were not present in more than 75% of the subjects, similar to what was done in previous studies. So this is the group structural connectivity network, which has a 65% of sparsity and the distribution of connections both for consistent and inconsistent connections. Functional connectivity was inferred from resting state fMRI data. After some typical preprocessing steps, we extracted the mean volt signal per region. Then we calculated the Pearson correlation between the signal of all ROI pairs to construct a symmetric connectivity matrix for each subject. All of them were then transformed to a normal distribution using Fisher's transformation to calculate the mean across subject. And finally, we transformed them back to Pearson to get the group functional connectivity network. Now let's move on to the methodology of metabolic connectivity. As I said before, the first 30 minutes of least mode paid data were used to reconstruct one image per subject. Then we extracted the FDG uptake from each region and normalized them by the mean FDG uptake in the gray matter to make this measure independent of the individual total uptake. So finally, we used a Pearson correlation between all pairs of regions across subjects as a measure of metabolic connectivity. And the last connectivity measure, structural covariance, was calculated from T1 images. After a visual quality control of the images, they were segmented into different probability maps, including gray matter, white matter, and CSF. Then we extracted the gray matter volume from each region, regressed out the total gray matter volume of the subjects, and calculated the Pearson correlation between all pairs of regions across subjects to obtain a group structural covariance network. After we had the 4 group networks, we calculated the similarity between the structural core and the other networks. And for that, we computed the Spearman or rank correlation between the connection weights, restricting the analysis to structurally consistent connections. So all these positive correlations are statistically significant because of the huge amount of data points, but the values go from weak to fair, being maximum for FDG covariance, what suggests that the structural connectivity network predicts better metabolic connectivity than functional connectivity. Additionally, we quantify the spatial overlap between the networks as a function of the sparsity level using the convergence ratio that is calculated as the number of common connections divided by the number of connections on average between the two networks, what would be equivalent to the dice coefficient. We adjusted the convergence ratio for chance to get rid of the overlap component that is not meaningful, and decided to restrict the interval to the maximum sparsity that still gives us a connected networks, as was done in previous studies. So it was 80% of sparsity in our case. These are the results we obtained. The highest overlap was found for structural connectivity and functional connectivity, 33% on average, followed by FDG covariance and gray matter volume covariance. In addition, we computed the similarity between the, let's say, surrogate measures of connectivity through Spearman correlations as well as spatial overlap over the same sparsity range. And both perspectives gave us the same ranking of agreement, being maximum for FDG covariance and functional connectivity and minimum for FDG covariance and gray matter volume covariance. And these values are comparable to those found on previous studies. Finally, we calculated the reproducibility of our results, repeating the analysis for a second scan session of the same cohort and in the same scanner, 8 weeks apart, and we obtained similar values and exactly the same ranking of relationships in the 2 time points. To summarize, I will say that this work systematically investigated associations between 4 connectivity indices measured simultaneously FDG covariant was associated with structural connectivity to a similar degree as functional connectivity, what supports FDG covariance as a valid index of brain connectivity. The low positive correlations and the limited proportion of overlapping connections indicate that each modality provides unique information, and the order of the similarity results is reproducible. As a take-home message, I would like to say that each connectivity measure captures a different aspect of the complex relationship between brain elements and, of course, has its own limitations, so we interpret the disagreement between them as a complementarity rather than contradiction, and we encourage the use of FDG PET to enrich our understanding of a human connectome. Thank you so much for your attention.
Since 2016, at the beginning of my career, I had the opportunity to model brain connectivity from FDG PET data. The study of metabolic connectivity may be challenging for some aspects. However, particularly important insights about brain system could be achieved by means of FDG PET. And today I would like to tell you something about that. Let's start with the biological basis of cerebral FDG PET. Glucose is the fundamental metabolic substrate in the brain. FDG PET reflects both oxidative metabolism in neurons and aerobic glycolysis in astrocytes, and is tightly coupled to postsynaptic activity. The FDG PET tracer is metabolized through part of the pathway of glucose metabolism. Unlike glucose, however, its product is essentially trapped in the tissue, allowing the PET detection and quantification of radioactivity. The bold fMRI signal is instead a semi-quantitative non-absolute index of neural activity. Indeed, bold signal is influenced by blood flow and blood volume and is also affected by vessel size. Thus, as opposed to bold fMRI, FDG PET can capture neural activity that is in steady state in our terms and is independent of vascular coupling. FDG PET connectivity studies have a long history. While PET connectivity studies make up about 2% of the literature about brain connectivity, its limited use is mostly due to its invasiveness. It's worth noting that some of the most relevant conceptual advances in the field of network neuroscience relied on PET. 10 years before the advent of resting state fMRI, several PET studies such as Horwitz and colleagues study examined FDG PET interregional correlation across subjects to extract between regions functional association. That is what we now define functional connectivity. 3 years after Horwitz's publication, Muller and colleagues referred to networks when discussing a linearly covariant pattern of metabolism between two or more regions. And it is from the study of activity at rest based on perfusion PET, these two reports were meta-analysis, that the FOMO network was formally identified in the early 2000s, and this advancement set the foundation for what we now call resting state networks. What about the methods used for extracting metabolic connectivity from FDG PET data? In most molecular studies, something like 90% of the study, connectivity is estimated from static FDG PET acquisition. Thus, the modeling is performed in subject series instead of time series, using for each subject the average signal obtained from the entire scanning session, with the idea that there is a meaningful structure in the inter-subjects variability. In this way, regions that co-vary across subject, namely their effect sizes going down and up across subject, can be considered as part of the same functional network. Various analytical approaches exist to examine the relationship between regions assessed by metabolic connectivity. Seed correlation or interregional correlation analysis involves picking a reference location, such as these regions of interest, and quantify the correlation with FDG uptake from these regions of interest to the other brain regions in a voxel-wise fashion. Thus, these steps allow for obtaining the connectivity map that depend on a seed of interest. The seed can be selected in either a data-driven fashion, so based on the data obtained by previous analysis in the same data, or on the basis of an a priori hypothesis. Another statistical approach is the principal or independent component analysis, which consists in a multivariate decomposition technique imposing constraints of orthogonality and statistical independence on derived components. The two methods, the principal and dependent component analysis, allow to identify coherent brain networks, for example the resting state networks, in a data-driven manner, without the need to select a specific seed ROIs in advance. However, the number of components to be extracted need to be set by the investigator. The selection of those components with pathophysiological and anatomofunctional meaning is thus crucial, discarding in this way statistical noise components. Two other approach that consists in the selection of several ROIs for the analysis of metabolic connectivity are the partial correlation analysis and the sports inverse covariance estimation, the latter also called graphical lasso. These approaches are due to widely used ROI-based approaches for connectivity analysis. The two methods compute a connectivity matrix starting from selecting a set of target regions. The partial correlation allows the estimation of the degree of linear association between each couple of selected ROIs, factoring out the contribution of all remaining ROIs. Instead, sparse inverse covariance estimation finds the estimated value of inverse covariance, thereby indirectly providing a measure of partial correlation. Once the full brain connectivity matrix is estimated through partial correlation of sparse inverse covariance estimation, graph theory indices can be eventually computed, molecular hubs and modules identified, and changes in nodal and global network properties assessed. These methods have been diffusely applied for the study of brain metabolic connectivity. For example, the first attempt to extract resting state networks from FDG PET data was performed by D and Visual in 2012. More recently, Savio and colleagues directly compared FMR and FDG PET resting state network and showed fair to moderate statistical spatial similarity between these two modalities. Still, some resting state networks were identified exclusively in the with the PET data, including subcortical network. And the differences found between the two modalities are likely due to the partly different aspects of neural activity captured by the two techniques and to the type of data used to extract their connectivity. It has the subject series in fMRI, PET, FDG PET series, and the time series in fMRI acquisitions. In my lab, the metabolic connectivity was applied to the study of different neurodegenerative conditions and also was applied to model the connectivity changes along clinical and/or neuropathological spectra. With this approach, we have demonstrated novel network-level alteration in a wide range of neurodegenerative disorders. In this sense, in neurodegenerative conditions, pathological events such as mitochondrial dysfunction, accumulation of neurotoxic protein species, and impaired neurotransmitter release produce a perturbation in glucose metabolism that can be reliably registered via FDG PET. For this reason, FDG PET is considered as a funnel biomarker able to capture all the different pathological mechanism events that may occur at neural level. So the combination of FDG PET measures with multivariate connectivity approach may provide crucial information about the spreading of the disease in neurodegeneration. With FDG PET, it is possible to track pathways for disorder propagation in vivo or to track neurotransmitter system known to be vulnerable in specific neurodegenerative condition. Together with the FDG PET, the application of connectivity approach has now been extended to other PET targets, including, for example, neurotransmitter systems. Topic that will be presented, however, later in the same symposium by Andreas Hahn. In recent years, several methods have been developed that allow the extraction of individual information from group-based connectivity matrix. These methods allowed the extraction of indices of network integrity for each subject and compare them between, for example, in these two studies, clinical cohorts. Moreover, to overcome the limit of temporal resolution characterizing FDG PET data, a recent study used high temporal resolution constants in fusion functional PET to measure subject level metabolic connectivity simultaneously with fMRI connectivity. However, further studies are needed to validate the reproducibility of this approach. To summarize, FDG PET is a reliable measure of postsynaptic neural activity, and the application of FDG PET to network neuroscience has a long history. Methodological advances allowed us to extract different connectivity measures from FDG PET data, and PET-based molecular connectivity adds unique complementary information to the quest for understanding brain organization. However, we still need to better understand the value of PET-based molecular connectivity measures, and that several steps are needed to fully establish PET in the field of network neuroscience. More studies are urgently needed to confirm and extend the findings using other analytical methods and other tracers, and address the effect of other experimental variables, for example, effect of sample size or sample heterogeneity on molecular connectivity results. Together with validation studies, derivation of individual metrics to quantify molecular connectivity alteration at single subject level represents a top priority in the field of neurodegenerative disease. Indeed, the availability of single subject metrics will allow to test the value of molecular connectivity as a biomarker of diagnostic and prognostic interest and to perform correlative analysis, for example, to investigate the connectivity metrics and their relationship with the other imaging and biological parameters or clinical and neuropsychological deficits, and eventually as a complementary outcome measures to evaluate the effect of new emerging treatments. Thank you.
Dear ladies and gentlemen, hello and welcome to my presentation about modeling brain connectivity using neurotransmitter PET. My name is Andreas Hahn and I'm from the Medical University of Vienna in Austria. The imaging modality of positron emission tomography offers the possibility to quantify neuronal receptors and transporters in the living human brain. This is a unique advantage which cannot be achieved with any other imaging modality. Conventionally, PET was used or is used for a regional investigation of the topological distribution of neuronal receptors. However, in this talk, I would like to present different approaches to assess interregional associations or connectivity with neurotransmitter PET data. This includes, for instance, seed-based analysis based on known neuroanatomical projections, as well as large-scale networks calculated across several regions of interest, and the combination with other imaging modalities. The first approach is a seed-based analysis. Here, a major advantage is to use well-known neuroanatomical projections of a particular neurotransmitter system and to include the specific information of the system obtained with PET imaging. For instance, for the dopamine system, neurons have their origin in the midbrain regions of the ventral tegmental area and substantia nigra and project mainly to the striatum or the forebrain. For a serotonin system, the neurons have their cell bodies located in the raffinuclei, from there projecting to the entire brain. In the raffinuclei, there are two major key players driving serotonergic neurotransmission. This is, on the one hand, the serotonin 1A receptor, which is expressed on the cell bodies, so it's an autoinhibitory receptor. And on the other hand, this is a serotonin transporter, which is responsible for the reuptake of serotonin into the presynaptic cell. So if the serotonin transporter is high in the raffinuclei, then the availability of extracellular serotonin is rather low because of the high reuptake. This will decrease the activity of the autoinhibitory serotonin 1A receptors, and as such the firing rate of the raffinuclei will increase. Presumably, this will also lead to an increased release of serotonin and, as such, availability of serotonin at the projection site. And this will also be followed by an increase in the serotonin transporter expression and a decreased serotonin 1A receptor expression in the projection site. So for both of these proteins, we expect an positive association between the raffinuclei and the projection site. So we can indeed test this association between the dorsal raffinucleus and projection areas with PET imaging, and I will show two examples, one for each of these proteins. The analysis is quite similar to that of a functional connectivity analysis with resting state fMRI data. The most complex thing is to extract your PET binding from the raffinucleus because the region is quite small. But luckily, in PET imaging, at least with these two proteins, we see the raffinuclei quite well. We can then do a voxel-wise regression analysis across the group and then get a connectivity estimate. So the first example is for the serotonin 1A receptor network. As expected, we found a positive correlation in healthy subjects between the raffinucleus and the amygdala and hippocampus, for instance. In patients with social anxiety before treatment, we found a rather similar pattern, but interestingly, after treatment with SSRIs, this association markedly increased, especially between the raffinucleus and the amygdala and hippocampus. So this underlines the importance of the serotonin 1A receptor in social anxiety disorder, and also that the treatment is dependent on the interplay across these regions. The second example is about the serotonin transporter network. Basically, we followed the same approach and rationale as outlined before. And again, we found a positive correlation between the raffinuclei and, this time, for the striatum in healthy control subjects. Interestingly, in patients with major depression, this association was markedly reduced, as you can also see here, and significantly different. So this makes a lot of sense because both the raffinuclei and the ventral striatum are important areas of reward processing, and anhedonia is a key symptom in major depression. So these two examples show that neurotransmitter systems are interdependent also across brain regions. The approach can also be used, of course, for other neurotransmitter systems, for instance for the dopamine system. And here you can use the seed region from the midbrain, as for instance substantia nigra or ventral tegmental area, and assess dopaminergic pathways. And here it is shown the high specificity when using the appropriate radioligand. In this case it was F-dopa, which is specific for dopamine synthesis, and there was a robust delineation of dopamine pathways obtained, especially when compared to the more unspecific estimate of glucose metabolism. The next step is to extend this to large-scale brain networks, similar to whole brain resting state fMRI analysis. Basically, the connectivity analysis follows the same or a common procedure. One defines an RI atlas and extract all the PET values for each subject, can then calculate an adjacency matrix with correlation, and in this case it's done again at the group level. We can then assess network parameters and compare these parameters between groups, for instance, with permutation testing. So there is quite some work available for the serotonin transporter networks, for instance in patients with attention deficit hyperactivity disorder. We observed a decreased association in the precuneus, and this may link default mode network dysfunction obtained with resting state fMRI connectivity specifically to the serotonergic network. Another example is in patients with major depressive disorder, and after 3 weeks of treatment with SSRIs, we observed strengthening of this network, particularly for the anterior cingulate and the insula, and interregional reorganization basically. We can furthermore define hub regions based on the serotonergic system and then assess connectivity across these regions. The last aspect I would like to cover is the multimodal combination of PET data with connectivity. The first example is about the incorporation of receptor maps in connectivity analysis in order to specifically link the resting state fMRI signal to the underlying neurotransmitter system. And this can be done in a two-step regression. In the first step, one aims to explain the resting state fMRI data specifically by the spatial distribution of the PET maps. And this results in a time course for each PET image, and one can then use these temporal dynamics in a second regression step, which yields the maps of the bold response, which are specific for each neuronal receptor. This is particularly useful for the assessment of drug effects, in this case, for instance, for assessing NMDA effects based on the distribution of the serotonergic network. So there were 5 different target proteins of the serotonin system included, and there were specific differences found for the serotonin 1A receptor and the serotonin transporter as observed here. So the beauty of this approach is that you can assess the functional response with respect to a specific neurotransmitter system. And furthermore, one can use average PET maps that are readily available at several databases. So there is basically no on-site PET machine required. One can go even more complex and combine structural, functional connectivity and molecular data. In the application here, the goal was to assess the effects of the serotonin 2A receptor on a neuronal model of LSD action. This is quite useful because LSD is a serotonin 2A receptor agonist, and the receptor was included in this model, as you can see schematically here and here. And it was shown that the distribution of the serotonin 2A receptor was specifically required to explain the variability in the LSD model. And this was particularly true as compared to different other receptors of the serotonergic system. To summarize, I hope I was able to show that neurotransmitter PET imaging is a quite useful tool for the assessment of brain connectivity. You have advantages of very high molecular specificity for a certain neurotransmitter system that cannot be obtained with any other imaging modality. This can be used, for instance, for the assessment of neurotransmitter pathways or for the assessment of pharmacological challenges, and you can enrich your functional connectivity analysis with databases that are readily available, for instance, with PET or messenger RNA data. Finally, I would like to thank the team from the Medical University of Vienna, as well as the funding institutions, and of course I would like to thank you for your attention. Please feel free to contact us if you have any further questions. Thank you and goodbye.
Hi everyone, my name is Joana Pereira. I'm an associate professor at Karolinska Institute and Lund University, and today I will talk about multivariate approaches to assess brain connectivity on amyloid and tau PET data in Alzheimer's disease. As most of you know, Alzheimer's disease is a neurodegenerative disorder associated with the abnormal accumulation of proteins in the brain, such as amyloid and tau. Several studies have shown that these abnormal proteins do not target random brain areas, but seem to accumulate according to a specific spatial pattern. In the case of amyloid, these deposits appear first in neurocortical regions and then spread to limbic and finally subcortical areas, whereas tau deposits appear in the transitornal cortex and then spread to prolimbic and neurocortical areas. Thanks to the recent development of both amyloid and tau PET tracers, it is now possible to assess these patterns in vivo. However, most studies have mainly used univariate methods to analyze these patterns and, for example, identify single regions showing greater amyloid or tau deposition in a group of patients compared to controls. There is increasing evidence showing that analyzing amyloid and tau PET data with multivariate approaches can provide new insights into Alzheimer's disease. So during this talk, I will show you different studies that have used these approaches to either assess the spatial patterns of amyloid and tau, their network topology using graph theory, or different models of disease spread to predict Alzheimer's disease progression. When it comes to spatial patterns, there are a lot of studies that have derived these patterns from tau PET images, and a few less also from amyloid PET. Here we have one of the first studies that assessed the tau patterns across the Alzheimer's disease spectrum. So in individuals that were cognitively normal, had mild cognitive impairment, and also AD dementia. They applied an independent component analysis, which decomposes the PET signals as a mixture of statistically independent patterns or networks. And interestingly, they found 5 different spatial patterns or components that were located in the visual network, the visual executive network, the working memory network, the precuneus, and also a typical network composed mainly of temporal brain regions. The tau PET signals in the first 4 of these components were negatively associated with age of onset of Alzheimer's disease, whereas the typical component showed a positive association. These findings are in line with early onset forms of the disease being associated with the typical tau patterns, whereas late onset forms are associated with temporal typical patterns. In addition, the authors also observed a progressive increase in the amount of amyloid pathology, cognitive impairment, and worse functional connectivity with higher scores in these different patterns or components. In another study focusing only on patients with Alzheimer's disease dementia, a higher number of spatial patterns were also identified using ECA. These patterns contain different brain regions from the temporal and occipital lobes, but also the posterior cingulate, precuneus, and precentral gyrus. To assess whether these patterns overlapped with resting state functional networks, the authors took the regions showing the highest tau in each pattern and used them as seeds to extract networks on functional MRI data from young subjects. And then they computed the overlap with the resting state network templates reported in the literature. Here is just one of the results they found for the precuneus tau pattern, which overlapped well with the seed-based network in young subjects using the precuneus, as well as the ventral default mode network reported in the literature. Interestingly, in a recent study, we were also able to show that these spatial patterns of tau are already present in cognitively normal older adults. In particular, we identified 10 different patterns, such as the medial temporal and perihippocampal, but also occipital, parietal, and frontal, most of which were symmetrical. We additionally found that these tau signals between the patterns were different between men and women, and some of them even correlated with memory and executive functions, suggesting that they might be useful to understand different cognitive phenotypes in aging. In addition to ECA, there are also studies using alternative techniques to assess the spatial patterns of tau and amyloid. For example, in a study conducted by Sepulchre and colleagues, a voxel-wise clustering was used to obtain the different clusters of tau and amyloid deposition in older adults. In this study, the tau deposits were grouped in tightly nested areas of the temporal lobe, and also atromodal areas, medial brain regions, and finally the sensorimotor cortex. While on the other hand, the amyloid deposits were clustered in atromodal areas and also in several patchy areas involving all the lobes. These findings support the notion that tau and amyloid accumulate within specific systems since very early stages of Alzheimer's disease. In a recent study by our group, we also applied a joint independent component analysis to identify the linked patterns in amyloid and tau that were made up of a combination of the signals in the 2 PET imaging modalities. In a mixed sample of cognitively normal patients with mild cognitive impairment and dementia, there were only 3 components in amyloid and tau that overlapped in similar brain areas, such as the left inferior occipital, the superior frontal, and the medial components. However, the rest of the joint components were actually quite different, indicating that amyloid and tau target different spatial systems, such as, for example, the medial frontal network in the case of amyloid and the temporal network in the case of tau. In line with this, we also observed in the same study that the amyloid and tau patterns overlapped with different resting state functional networks, with amyloid mainly overlapping with the networks that belong to the default mode network, whereas tau overlapped with a wider range of functional networks And we also observed that whereas tau correlated with different cognitive functions, the amyloid networks only correlated with memory. And finally, also within the same study, we observed that in contrast to amyloid, the tau patterns also correlated with anatomical connectivity. As you can see, these different plots show the correlations between tau signals within each of the previous patterns and reduced fractional anisotropy values in different white matter tracts. When it comes to network topology using graph theory tools, there have been a few studies employing this approach to understand the relationship of amyloid and tau between different brain regions in Alzheimer's disease. In one of our studies, we assessed the amyloid networks in the individuals that were in different stages of amyloid accumulation, defined by cerebrospinal fluid and PET amyloid measures. To build these networks, we first extracted the amyloid SUVR values from different cortical and subcortical regions and estimated the connectivity between these regions using a Pearson correlation analysis. One of the main findings of this study was that subjects that were in early stages of amyloid accumulation had a higher number of connections in medial brain regions such as the precuneus and posterior cingulate. These regions overlapped quite well with the areas of the default network, which is in line with previous studies showing that amyloid pathology preferentially accumulates into regions of this network. In a more recent study, the network-based relationships between amyloid and tau accumulation in older adults were also assessed. So in this study, the authors created a weighted degree map of amyloid to tau correlations and also of tau to amyloid correlations. And they found that there was a higher weighted degree in the parietal cortex, the precuneus, the orbitofrontal cortex, and also superior frontal gyri in the amyloid to tau correlations, whereas in the tau to amyloid correlations, the results were less widespread and stronger in the inferior temporal gyri as well as the precuneus. When these weighted degree maps were correlated to cognitive performance, interestingly, the only one showing results was the one that comprised the amyloid to tau correlations and was observed across different areas of the frontal, parietal, temporal, and even the occipital lobes. These findings indicate that specific correlation maps of amyloid and tau are closely related to cognition. And finally, in a study conducted by Iacarino and colleagues, correlation networks were also built for amyloid and tau, in addition to gray matter volume within the same individuals. In this study, the authors found that amyloid was mainly associated with distant gray matter regions between lobes, whereas tau was associated mainly with neighboring gray matter regions within the same lobe, suggesting that amyloid might have a more distant effect on neurodegeneration, whereas tau has a more local effect. An area that has become the focus of recent research is to combine the multivariate analysis of amyloid and tau PET data to build models that can predict disease progression and the spread of pathology in the brain. Using a novel approach called directional graph theory regression, in this study, Sepulcher predicted tau and amyloid pathology changes in older adults, both cross-sectional and longitudinally. At the cross-sectional level, they found that tau deposition in the temporal region predicted tau in other parts of the temporal lobe, as well as medial frontal regions, whereas amyloid in the posterior cingulate predicted amyloid in frontoparietal regions as well as lateral temporal areas. At the longitudinal level, they found that tau in this temporal region was associated with the spread of tau to the precuneus and posterior cingulate, whereas amyloid propagated to similar regions found at the cross-sectional level. So altogether, these findings show that amyloid and tau have distinct propagation pathways in the brain. And in the same study, they also found that these distant propagation pathways were associated with different genetic profiles, which were more axon related for tau, which was associated with the MAP gene, and more dendrite related for amyloid, which was associated with the CLU gene. Recent studies have also started including information about brain connectivity in order to predict the spread of tau pathology in the brain. In particular, in this study, the authors tested whether functional connectivity was associated with future tau accumulation in Alzheimer's disease. And they found that future tau accumulation could be modeled by a combination of baseline tau levels, functional connectivity, and distance between brain regions, which is represented by model 3 in this figure, which obtains significantly better results than the other two models included in the study. These results were further confirmed in 2 different cohorts, supporting the view that tau transmits pathologically across neural connections in Alzheimer's disease. And finally, in a study that included both anatomical and functional connectivity, to test the hypothesis that tau spreads through the brain's connections, they used an epidemic spreading model to simulate tau spread and compare the simulations to observe patterns in 200 individuals across the AD continuum. In this study, they found that the prediction model that included anatomical connections derived from diffusion tensor imaging overlapped much better the observed pattern compared to the model that contained only the functional connections, although this model was not bad either. But this overall suggests that tau may actually preferentially propagate through the brain's anatomical connections. So to conclude, in this talk, we observed that amyloid and tau accumulate across different spatial patterns in Alzheimer's disease, which correlate with worse cognition, overlap with different resting state functional networks, and in the case of tau, correlates with worse anatomical cognitivity. The studies assessing network topology show that changes in amyloid networks seem to occur in areas of the default network. The interaction between amyloid and tau correlates with cognitive decline, and amyloid and tau seem to have distant and local effects on gray matter volume, respectively. And finally, studies using spreading models have shown that amyloid and tau have distant propagation pathways that are associated with different genetic profiles. And tau seems to spread through functional, but more importantly, through anatomical connections. I would like to thank Ariana Sala and Igor Yakushev for organizing the symposium, and of course, my group collaborators in funding. Thank you for listening.